have all the AI computation speakers here, please? Away you go, Tom.
The microphone is on. Thank you. So thanks all for a great session and undoubtedly a bright future. But I have a very specific question for Jane. And first of all, I want to pay you a huge compliment for having the courage to set up and explore the platform that you described today. I realized the huge risk at the beginning of whether this was going anywhere or nowhere. And it's very gratifying to see that we're seeing some deconvolution going on and targets coming up. So fantastic. And another compliment that you go into neurodegenerative diseases, which is, about the most difficult area to go into with antibodies, but you do it anyway. So as far as I'm concerned, and if I have anything to say with it, you're invited to next year's TAS 24 meeting, because some stories are worth telling every year, and it felt a bit like a Netflix story. You're into it, and then you wait for the next episode. Yeah. It takes too long to air anyway. All right, that's enough for that. But I have some specific questions, because if you take a helicopter view of all the targets that you actually have identified, what is your feeling? Are these complete surprises in terms of their correlation with disease, or is it more the functionality of the antibody that you finally find to say, oh, this is really different from anything we've done through other approaches?
Yeah, so first off, thanks, Tom, for the support, and the Netflix analogy is quite nice. The targets that we've been finding, we do find clinically validated targets, as I said. The sweet spot that we're looking for is targets where we've got a level of confidence, maybe from beyond the resilience association, that there's some other sort of signal that suggests, yes, this is relevant. So we do try and look for genetic associations of the targets in the diseases that we've been digging into. So CD33 is an obvious example, but for the other target that I couldn't name there is a sort of SNP association, so some more obscure genetics. So I think we sort of use that as a bit of a hurdle to sort of bring things into the validation phase. And then we just have to do really hard work on functional screening and try and work out what's going on, and do we think that's a relevant pathway in the disease. We've had a few false starts, not surprisingly, but it's one of the hardest things to do, is sort of choose of our target landscape, where do we think we're in that sweet spot of novelty, but with a little bit of validation. So if that answers the question.
Yes, sure. And maybe two short questions, because I don't want to clog up the microphone, but do you feel like there's one antibody species that does it all in a patient, or you suspect that there's other things going on?
No, that's a good question. And ultimately, we'd like to start looking at, are any of these convergent sequences seen in pairs? Actually, for the pancreatic cancer, I'm not sure whether it was. We certainly, in oncology, we have seen anti-PD-1 and anti-CTLA-4 in the same patients convergent. I suspect that there will be antibodies that are jointly convergent in neuro. We just haven't had the kind of tools to really investigate that. But I think that's the sort of next wave of what we need to do, because I think it's a bit naive to think, well, one antibody is going to solve. So we'll see. But at the minute, we're keeping it simple and just assuming that one can do something. Yeah.
Okay. Well, looking forward to the next episode for sure.
Thank you, Tom.
So thank you very much. I also was very interested in your approach. And indeed, you indicated that, sorry, this question is for Jane, that you're sequencing approximately a million sequences per individual, right?
That's the top end, yeah.
So when I was trying to make kind of a back of an envelope like calculation, so it seems like the human genome is considered, human repertoire of antibodies is considered to be approximately 1 trillion antibodies, right? So 10 to the 12th. And if you are sequencing 1/millionth of that,
It's still tiny.
That means that it's a very, I mean, you're sequencing a very small number of actually antibodies. And then you're sequencing this amount, a million of antibodies in each patient. So the probability to find the convergence in more than one patient implies that these are hugely overexpressed antibodies and events, and therefore maybe you don't even need to worry about this convergence. You just look at the highest expressed antibodies. And so, but the real question is, do you actually see that in that million, the convergent antibodies actually represent that very highly overrepresented species?
Yeah, so we look at the representation in the sequence library. Now, that doesn't necessarily correlate with the production of the antibody in serum. We're actually building a lot of serum profiling capabilities as well, which I haven't had a chance to talk today. But I think that's the best way to answer that question of, are we finding very overexpressed antibodies in serum? But the surrogate for that we use is how big are the clonal, how many times we're seeing that clone in the million sequences that we find. I think it is, the probability is really interesting. So actually, I think the significance of finding convergence, given that we are sampling relatively small numbers, makes it more significant but the deeper we sequence, hopefully, the more we can explore that. There's around 10 to the 9 BCRs at any one time in any individual. So I kind of think sort of it's 10 to the 9, 10 to the 9 rather than 10 to the 13, because that's what we've got in the body at any time. But yeah, it's very hard to put statistics on it. I mean, the thing is, the convergence that we're finding is clearly some antibodies that are being actively selected. They're mutated, they're class switched, and they're continuing to evolve. So I think that gives us confidence it's a real biological effect as well. Yeah.
Thank you.
I also have a question for Jane, actually. So there's this Mylotarg. It's anti-CD33, very powerful ADC. Of course, there's blood-brain barrier, but I just wonder if you were thinking, looking at these patients, what would happen to them in terms of Alzheimer's disease? I don't know. Yeah,
I mean, I think there's not a lot of data on what's happened to any patients that have been treated with anti-CD33s in an oncology setting. I think it's an interesting thing to think about. Yeah, it's a good idea. Yeah.
Okay. Yeah. So I have a question for Brian and for Jane. A very nice presentation, all of you, I have to say. I'm intrigued already for over 25 years on having a light chain pairing, and that's sort of my question then goes back to work I did myself 25 years ago on autoimmune libraries. And when we select for an autoantigen with an IgG repertoire, you typically get many mutations in the heavy chain and also many mutations in the light chain. If you select to the same target with the naive IgM library, you get usually a heavy chain with limited mutation, also the light chain with limited mutation. So you showed, Brian, sort of, your distinction in V genes and somatic mutation. So what we also, and so are you using that information in your models? And second thing, so that also realizes if you then for an autoimmune do a light chain shuffling, what Jane is routinely doing, I understand, light chain shuffling to optimize your antibody, to find your pair. There, if you have an antibody with many mutations, you very often find only less than 1%, or if you have to do phage display to find the light chain back that shows the same binding to the target. Well, if you take in non-immune library from an IgM, you find many light chains, let's say 60, 70%. They support the same antigen binding. So just like to have your thoughts about if you're using that information or in your models, or just a thought. I don't know who wants to start. But.
I'll start? Okay. So in terms of training the models, we're not giving the model any information beyond the sequence. Okay. So it's learning that essentially de novo, right? It's somehow figuring out that there is such thing as a germline and there's such thing as mutated, and it groups them accordingly. But yes, you're absolutely right. If it does appear that correlation holds very well in natively paired sequences. So if we're doing some sort of light chain shuffling or doing something different where you want to either infer or try to repair heavy chains with light chains or vice versa, you should be roughly matched in that direction. So yeah, I agree.
I'm not quite sure what I can add to that, but just to say, we do see a relative promiscuity in our light chain models, our pairing models. But yeah, we see sort of consistency in sort of target binding. And just a sort of word of caution on, because our heavy chains are selected in a particular way, I actually think we're probably biasing our heavy chains for the CDRs in the heavy chains to have more impact on binding than the natural repertoire. So I think we just need to be sort of wary about comparing the approaches because of that. I mean, I haven't got evidence for that. It just seems intuitive that that could be the case.
But let's say if you do a shuffling with your light chains, do you then find that many light chains support still the binding, or is there really a limited number of sets which support your binding to the target?
We find quite significant numbers of light chains. They are very more promiscuous than the heavy chain. So yeah, we see a range of light chains giving what looks like the same functionality.
Thanks.
Just add up one more small thing. So Victor mentioned when he was that mutations tend to be deleterious. More was it Victor or was it one of you? Mutations tend to be deleterious more often than not. So if you're trying to find a light chain, it might actually be easier if it's unmutated because you have such a high likelihood of breaking it by incorporating mutations too. So yeah, I think that.
So I was thinking, if you have many mutations, then maybe the structure of your heavy light chains is much more fixed, and there's less freedom for your light chain to pair. While if you have a germline, you're flexible, and maybe your light chain is also more flexible. I don't know if that's correct, but. Right, thanks.
One question for Jane. So, since you're really focused on neuroscience drug discovery, in putting your platform together, have you selected for yourself a Trojan horse domain so that you're fully accessible to the CNS with your antibodies?
Are we using any blood-brain barrier kind of transport? Yeah.
Of course.
Yeah. So it's a good question. At the minute, not. Well, we're sort of building an option to have one in-house in case we need to. From the data we've had so far, 3 of those antibodies are in vivo models, and we seem to be seeing some effect without the need for any transporter.
So you gave the idea that with the CD33, you had peripheral access to the microglia, and so that made some sense, even though I don't know that biology fully. Yeah. But for the others, especially say for your target Huntington, that's a totally different.
Yeah, it's totally different. But I think again, I mean, a lot of the amyloid antibodies that are already out there don't have transporters like.
But we know why that works.
Yeah,
because you're depleting, you're in the periphery depleting, and you pull it out. So that we know how that works.
But I mean, we've done a lot of PK analysis on the neuro antibodies, and we're seeing, between 0.1, 0.3% going into the brain just in the sort of normal mouse models. So we'll just have to wait and see. I think if we need one, we'll put one on.
So the other, like a very hard question is that, as I understand it right now, Huntington still has no known function, and yet you've identified a candidate which targets Huntington. Does this candidate's putative epitope therefore give you some hint as to what the function may be?
Yeah, so this is, I didn't have a chance to talk about that program, but we found, convergence for antibodies that recognize the exon 1. And what we think is happening is we are recognizing extracellular Huntington, which is interesting. So we think we're blocking the extracellular seeding of the Huntington. And by doing that, we're not affecting the normal Huntington within the cell. So we think there's a potential differentiation there from some of the other approaches where they, targeting Huntington and potentially knocking down the normal function. I think that's a big risk.
Were you selecting from extremely expanded people who converted later phenotypically? I mean, that's really interesting.
It's a sort of long story on the Huntington. We actually found it in broader neurodegenerative cohorts, and we're kind of now mining. We've got collaboration with CHDI to see how that translates into the patient population. So.
And for Dr. Bryony, so you give me the impression that with a binary classifier, you're good for 1,000 antibodies to a given target to get to 70% to get a good binary classifier. So theoretically, instead of trying to put everything into one model, would it make sense for you to take your 40 respiratory viral models and train 40 different ones and then run your patient's single antibody sequence against those? Statistically, where am I if I were to try to do that?
Yeah, that's actually a pretty well-known approach. People call it mixture of experts or other things, where you have multiple models, and you can even train a model on top of those models that picks which model is the best predictor of things like that. So yeah, it's not immediately clear that a single monolithic multi-class classifier is the best way, or whether multiple individual models is best. Yeah, that's something that we'd have to kind of empirically determine.
Hi, so great talk from all of you, and also thank you for challenging the concepts, moving the field forward, and also to be such an inspiration in the end. So I have a question for Shirley. So also keeping the mood on the listen to the tumors, have you checked the expression of the target, the IGS-F8, according to hot and cold tumors? Have you tested that?
Yes, so IGSF8 is higher expressed in immune cold tumors.
Great. And also, in terms of the receptors, you mentioned at the end of the talk that there was a difference between the receptors on the NK and the DCs between human and mouse. Have you also checked the correlation of the receptor expression with the different crosstalks of the NK? And I guess what I'm thinking behind is, like, for example, the crosstalks in the vaccine context, where you have the memory CD4s helping and improving NK cells function. Have you kind of translated a bit or checked that in the context of the tumor, where we have a persistence of the antigen? In the end, the question is any modulation of the receptor according to the different crosstalks of NK cells with other cells?
Yeah, so I think that so the ligand and receptors are kind of correlated, but not the antibody with the receptor so far. I don't think we can see strong correlations there.
Okay,
yeah.
Thank you.
Right. I also wanted to compliment everyone on phenomenal section, but given that we are getting to the end of the day, right, I wanted to limit to one question, and we'll stay with the oncology for this question, right? So you looked at all types of antibody sequences, right? And I really loved your analogy about looking at the pictures taken in different zoos. But obviously people also take pictures of themselves, of pigeons, right? So it's not necessarily a relevant picture taking. There are a lot of B cells that are just in blood vessels, right? Which are obviously contained in the sequence of the total tumor. But now you have focused on antibodies that are present, you know, the most correlate to what is actually present on the tumor mutational status, right? What you expect to be expressed or overexpressed, right? So how obvious is from that picture that IGSF8, I might be wrong in pronouncing the target name, right, is the top target? So can you sort of walk us through how many candidate targets were discovered, right? And where was the target you chose in that selection? And what was sort of the definitive decision-making point that this is the target to pursue for a company?
Yeah, very, very good question. So as I mentioned, you know, we first focus on IGSF family because it's kind of known to mediate cell-cell interaction for IO target. And also we look at the CRISPR screen result and we say, oh, there's a hit. And then it's overexpressed in tumors, and then that's when we went ahead. And also we look at the patent database. There are actually good targets. For example, TJ actually scored very high on our target list as well. But then it's so crowded, so we didn't develop or test the antibody, the prediction there. And so we try to look for something that's not so crowded and it has a phenotype. At least the gene has a phenotype in CRISPR screens, overexpressed, and then we tested this antibody. And when we saw the in vivo efficacy, we still didn't understand the mechanism, but that efficacy gave us the confidence to work backward on the mechanism. So right now we are doing this for additional target, and some of those we do have the functional assays established ahead of time before we get the binder. So I think, I think in some sense, we are certainly lucky in one, but we are looking at additional ones as well. So it's kind of a combination. So I would say the AI model is certainly not perfect, but it gives us an orthogonal approach to rank target. For example, Rick Young published papers to show that super enhancer, from H3K27 acetylation can give you some target ranking, right? And they have a company on this as well. By that alone, it can give you some idea, but you also need additional functional genomics data.
So is it fair to say that you limited your choice of targets first, right? And then looked into the tumors, whether you can actually find antibodies that would bind to those?
So right now, we have some additional criteria, say cell-specific expression, at least that is higher in some cells than others. We also look at the expression in most of the normal tissues, and we also examine the evolutionary conservation is actually important. Also survival information, immunotherapy trial indications, and also evolutionary conservation, because we want to get an antibody that potentially can, we can do in vivo studies. And so we have a kind of a tiered criteria to do something like this.
Awesome. Thank you.
Thanks.
Okay, final question.
This one will be quick. Okay, and it's easy in some regards. And Samir, thank you guys, and Brian, thank you guys so much for all the contributions you've made to our field. I mean, these are tremendous. I think we probably owe you back in kind, maybe in some sequences. So, in thinking about the protein structure model prediction versus protein folding prediction and the incorporation of physics, how is that going to change the way our computational approaches to understanding protein molecular dynamics? How do you see that changing going forward?
Okay. So the excellent question, because I forgot to mention that AlphaFold hasn't actually solved the protein folding problem. What AlphaFold has done is prediction of protein structure. We still don't understand how proteins fold, and there is no physics involved in any of the process that AlphaFold has. Same goes for the language models as well. But I think what it does learn is packing and how different packings work and what environment they work. That's what it learns, both in the case of language models as well as AlphaFold. And this is why it's good in predicting domains. And in many cases, you would find individual domains have very high confidence, but the interaction between different domains are not predicted with that high confidence. Now, having said that, it extends very well to complexes because the interaction in interfaces is very similar to interactions between domains as well. So we don't have different physical properties for domain-domain interaction versus complexes. And this is why it extends. Now, there are many new models that are being developed where they are incorporating some of the physics and with the idea to understand conformations, etc. So that work now is starting, and AI will play a role in that in terms of also making things faster. So if we want to have a simulation, you would have to have a simulation long enough, which means a lot more compute, to really get something meaningful out. And there, AI could help in terms of making it faster in identifying which paths to take to get to the right confirmations to get maximum information out. So there are two more kind of problems here. One of the problems is we can generate multiple structures, and there are many different ways of generating multiple structures. But we still don't know if the structure we have generated actually is biologically meaningful. So that's one problem that is difficult to answer. The second problem that is difficult to answer is what should we actually study? So now that we have all this huge amount of information, which bits of it should we take forward in terms of either developing new methods or studying it experimentally or studying it computationally, which will give us maximum information to then understand more about biology. And these two kind of, there are ways in AI to understand which portions of a latent space could give you maximum information content. But those are the kind of things maybe we should be also thinking about to limit the number of experiments we have to do to take the next step. I don't know if I answered all your question, but that was very and just remember, protein folding problem is still not solved.
Okay, just hold that microphone for a sec. So I just want to ask you one question, and then we're done. So, do we need crystallographers and EM people anymore now? And what about in 5 years' time?
So we do need both crystallographers and EM people for the reason that AI is still a tool, and as I said, these are predictions, and so we still need validation. Crystallography still has...
If your predictions are 98% correct, that's good
yes,
enough for me. That's good enough for me.
So we only know they are 98% correct in the cases where we have the experimental structure. And we now have 214 million predictions. So that's one thing. Second thing, as I said, the domains may be correct, but not the interactions between domains. Complexes and dynamics is still an unsolved problem. For small molecule related drug discovery, X-ray still is one of the best techniques, although EM is catching up. And EM is going in the direction of in situ structures, which is going to give us a lot more in-context information. And there is no other technique that would give us that kind of information. So EM is actually going to give us data that will require us to have new AI methods to do segmentation, to identify those proteins, their locations, their conformations, what they are doing, etc. So there is huge amount of experimentation that needs to happen. So when AlphaFold came out, the structural biology community was in shock. But very, very quickly, I think everyone has realized that this is a tool, this is not the answer, and we still need a lot of experimentation to generate new data to validate the existing models.
Great answer. Okay, that's a great answer. Great session. So I think the only thing to say is 8:55 in the morning tomorrow. Start. Thank you. Thank you.